Hello, let's talk fermentation.
Alright, in order to understand what fermentation is, we must know what glycolysis is. Glycolysis is a catabolic process in which glucose is broken down into two molecules of pyruvate. This is the first step of aerobic cellular respiration. But what happens when there is no oxygen? The cell still needs to keep making ATP because it is a source of energy. But how does the cell do it? Basically, glycolysis keeps going. That's the way the cell keeps producing ATP. But in order to do this, the cell needs to regenerate NAD+. We need to understand that NADH doesn't come out of nowhere. Basically, we have NAD+, which is reduced into NADH, and two molecules of ATP are produced. This all happens during glycolysis. Now, let's understand something. NAD is reduced to NADH because it is gaining electrons. NADH is oxidized into NAD because it is losing electrons. During glycolysis, NAD is reduced to NADH. However, during fermentation, NADH has to be oxidized to NAD in order for glycolysis to repeat itself and in order to produce ATP without the presence of oxygen. In other words, we are regenerating nicotinamide adenine dinucleotide during fermentation. There are two different types of fermentation: alcoholic fermentation and lactic acid fermentation. During alcoholic fermentation, the two molecules of pyruvate produced during glycolysis are used. These will produce carbon dioxide and 2-ethanol as a waste product. The derivative of pyruvate, which is acetaldehyde, can function as an electron acceptor. In this way, NADH is oxidized into NAD+, and glycolysis can repeat itself. Alcoholic fermentation is carried out by yeast while making bread. Carbon dioxide helps the bread grow, and the alcohol produced is evaporated while baking. The second type of fermentation is lactic acid fermentation. Lactic acid fermentation can be carried out by your muscle cells if they are experiencing oxygen debt, for example, when you work out intensely or when you run a lot. Lactic acid fermentation starts off the same way as alcoholic fermentation, with the two molecules of pyruvate produced during glycolysis. The 2-pyruvate will yield or produce 2-lactate. However, how are we going to get our NAD+ back? Well, in this case, pyruvate is going to be functioning as the electron acceptor. By doing this, NADH is once again oxidized into NAD+, and glycolysis can keep going. Now, let's review. During fermentation, cells need to find a way to regenerate NAD+. By doing so, NADH is oxidized into NAD+. This occurs because NADH loses its electrons. Even though fermentation is pretty cool because you can produce ATP without using oxygen, the production of ATP will always be greater when oxygen is present. Thank you for watching, and remember,
This is Miriam, over and out.